Is Wendy on yet? Oh, there's Wendy. Okay, great.
Hello.
So it's 3 o'clock, so I think we should get started right away. Very excited today. Welcome to the inaugural Discovery to Commercialization Speaker Series. And a special welcome to those of you who have joined from across the country and from other countries for that matter. We have a stellar lineup. For those of you who don't know me, my name is David Granville, and I'll be your host for this monthly speaker series. But before beginning, I would just like to acknowledge that in BC, we must also recognize that we are conducting our business today from the homes and offices on the shared and traditional homelands of the Musqueam, Squamish, and Tsleil-Waututh peoples. So the way this series got started, the Faculty of Medicine developed a comprehensive strategic plan a number of years ago, and one of the foci on this research was on the research pillar was focused on bolstering innovation, commercialization, industry partnership, and investment, and I was asked to help lead this endeavor. So over the subsequent months, we established a working group tasked with identifying areas in which we could do more in the Faculty of Medicine to improve innovation and commercialization while improving our relationships with industry and in order to combine forces to combat health challenges such as what we have today and to remove the us versus them, so to speak, mentality that has often plagued relationships between academics and industry. So upon identifying a number of key areas, we then met with key leaders, about 40 key leaders from small, medium, large biotech companies, hospitals, health, life sciences, BC leaders, and academic entrepreneurs from the early stages of wondering whether they should commercialize something to those who had done it many times before. And some of the key themes to arise from this workshop were around better educating faculty and trainees in this area, not only to improve the success in innovation and ability to take discoveries into practice, but also to help to train the next generation for the jobs that are in need currently. And other areas that were identified, which we'll hear about more in later times, were around breaking down barriers between collaborating between industry and investment, asset mapping and how to navigate the Faculty of Medicine, improving communication, faculty mentoring, incubator space, clinical trials, alternative funding models, leadership and training. So there are many wheels turning, but the goals of this seminar series are to basically start bringing people together, and perhaps there's no better example recently of academics, industry, and health professionals coming together to solve a problem than the pandemic we're facing today. So the general structure of this monthly seminar series is going to be identifying a clinical leader to state the problem from the clinical perspective, come in with a scientist to sort of discuss some of the most recent findings in late-breaking science, and then come in with a couple companies that may be coming up with solutions around solving the problem. And then at the end, we would like to incorporate in another aspect of the strategic plan around some of the cores that we're starting to develop and start spreading the word and advertising on some of these core technology areas that we're doing. A few housekeeping rules or notes, I would say. So this event is being recorded and will be posted on the website. If you have any questions, please put them in the chat box. A moderator will lead and answer the questions, and please remain muted unless you need to speak. So this will minimize the background noise. And just here before, in case I don't mention it at the end, I just wanted to send out a huge thank you to the people that helped put this together. So Kara Colazar, Amanda Chisholm, and Vivian Sum. So virtual flowers to you three for all the hard work that you've done in pulling this together. So at this point, it's my pleasure to introduce our two moderators. For the first two talks, Wendy Herbert will be moderating. She's a senior executive who brings more than 20 years of global finance, operations, and compliance leadership experience in healthcare and technology sectors. She also has she's had key leadership positions in J&J, the Heart and Stroke Foundation, and Lexmark International, and holds a BA from University of Western Ontario, as well as an MBA from Queen's. And the second moderator, Dr. Deborah Money, is a professor in the Department of Obstetrics in Gynecology and Medicine and the School of Population and Public Health at UBC. Deborah is a subspecialist in reproductive infectious diseases and also has done a lot of work with COVID herself, and an immediate past executive director of the Women's Health Research Institute, past vice president of the BC Women's Hospital, and past executive dean for the UBC Faculty of Medicine. So at this point then, it's my pleasure to pass the microphone over to Wendy to introduce our first two speakers.
Thank you very much, David. I think this is going to be a great initiative. As you say, nothing more timely than bringing together all of these stakeholder groups that make our life sciences sector so strong and exciting in British Columbia right now. So it's my pleasure to introduce an extraordinary leader within our BC community who also recently was awarded with the Order of BC. So I'm quite pleased to be able to talk about that. But our first speaker is Dr. Mel Chan, Medical Director of the BC CDC. I think he probably doesn't need much more of an introduction, as he's so well known and has really been the face in leading the COVID-19 response for our province. So thank you for your leadership. And I will turn it over to Dr. Mel, as I like to refer to him.
Thank you, Wendy and Kara. So thanks for inviting me. And so I'm just going to talk about where we are from a public health perspective and try and do it quickly in 10 minutes. Next slide. So if you look at the figure here, and you're the person on the left who's infected versus the figure on the right who's not yet infected, the data suggests that most of the infections are due to contact or droplets, and aerosols can be a factor, and the droplets usually don't go beyond a meter, but aerosols can go beyond a meter. Environmental fomites are not felt to be an important source. And basically the points of entry of this agent are eyes, and the eyes, the lacrimal duct goes into the nasopharynx directly, nose, or the mouth when you inhale it. If you ingest it, it probably would be inactivated, but no one has data on that. The important thing is that there's a lot of literature on people picking their nose and having eye contact approximately 15 to 25 times an hour. Next slide. So the data supports the fact that your transmission risk is reduced when you use personal protective equipment. And this is true whether it's SARS, COVID, or MERS. And the three important components are physical distance. And physical distance is more than just physical distance. It's about airflow. It's about dispersion. And in this figure from a report by Chu et al., which summarizes the literature, essentially, most of the time, 1 meter is probably enough, and 2 meters may be necessary when you're in a confined space. Very difficult to message from a public health perspective, and you can see that in all of the stuff going on in the media. Face masks. There's clearly a gradation in mask quality, with N95s being the best, but if you have to wear one for a long time, it's really difficult. Surgical masks being next. And one thing they do is prevent you from picking your nose, though a lot of people wear the mask under the nose, which is not a good idea. I'm going to show you some data on its impact on dose, and that may be important. Eye protection is very important. If you look at healthcare workers, properly using PPE, most of their risks occur from their home or their transit environment, not from working. Next slide. This is a very interesting paper by Gandhi et al., and it talks about reducing the inoculum of SARS. And if you looked at the Diamond Princess, which docked in Yokohama in February, There were 634 people on board, and about 18% of them had this asymptomatic infection. We'll talk about that in a minute. In March, there was an Argentinian ship with 128 people on board, and 81% of those were asymptomatic. On the Argentinian ship, surgical masks were issued to all passengers and N95 to all staff. In some outbreaks in an Oregon seafood processing plant, in the Tyson chicken processing plant in Arkansas, face masks were required, and essentially most of the outbreak cases had asymptomatic disease. And sometimes they don't admit that they have symptoms, but I think that's a very important thing from this literature, suggesting that there is a relationship between masks, transmission, the infectious dose, more dose, making a more intense infection, and symptomatic disease. Next slide. In terms of diagnostics, most of the nucleic acid testing that's available in the country has a sensitivity in the range of about 90 to 95%, and a very good specificity in the 99.5%. It detects the viral RNA, and nasopharyngeal swabs are the sample of choice. Throat swabs are a tad less sensitive, and there are, as you all know, global swab challenges, and there's been a push to use saliva, and we've recently rolled out saline gargles for our student-age kids. It's important to get the turnaround times less than 24 to 48 hours, because if the result is delayed, then it basically drives people crazy. And the other thing you should realize is that as you deal with people who are less likely to be infected, you will get false positives, probably at a rate of 1 in 200, largely due to technical errors, not that the test is picking up another virus. Most of the transmission, based on some publications by many al in Nature Medicine, are likely due to symptomatic infections and their contacts. But you clearly can have pre-symptomatic and asymptomatic infections, and we just discussed the scenario for that. The tendency is to try to use testing as a mechanism to open up society. As you can see from across the country, this is not trivial to do. There are antigen tests which are at this point not yet available in Canada. They're a little less sensitive, and the question is do you test someone multiple times with a less sensitive test to open up society? That's a complex issue. Next slide. Genomics. So in terms of genomics, the virus mutates slowly, but if you look at this paper from Cell, the G614 has replaced the D614 as the dominant strain. So the question is this truly because of a change in the fitness of the virus, or is this just a function of the fact that China shut down most of its transmissions and most of the global transmissions actually emanated later from Europe? And so there are a lot of questions as to whether it's more infectious or not, or less lethal, and whether there's a change in neutralization. There was a recent report in Lancet of a Delta 382 deletion variant, and it said, oh, this is less virulent. But if you look at that paper carefully, much of the people who had the mutant were much younger. And as you all know, the literature supports age is the most important predictor of poor outcome. Next slide. Serology is a mess, and largely because of the complexity of it. If you look at the commercial lab-based systems, they're approximately 95% sensitive, 99.5% specific. Point-of-care tests occur. They're a little less sensitive, but so far many of them have been inconsistent in performance. In terms of their uses, they have limited diagnostic utility. The reason being that by day 7, only about 30% of people will be seropositive. By day 8 to 14, 72% approximately will be seropositive. By day 15 to 21, 91%, and approximately 95% by day 21 to 35. That means if you don't diagnose them, they're either better or, shall we say, not in good shape. It is helpful to diagnose people with multi-system inflammatory syndrome in kids, and that is probably one of the more important uses at the present time. Initially, the push was to assess for seroprotection. There's monkey models that support seroprotection. There have been at least 5 human reinfections that I know of at this point. In some cases, the initial infection was mild. Sometimes it's stronger after the reinfection. And so we're going to study all of these issues. And what has been done with these assays is to determine seroprevalence. And essentially, that data suggests that somewhere between 1 to 10% of Canadians have been infected. And there may be higher proportions of people infected in some subpopulations, but the importance is there's many, many more people to infect. Next slide. This is what I'd like to spend a little bit of time because it's very important. There's this concept called original antigenic sin. It was described by Thomas Francis in 1960, and it's described as a hierarchical antibody response after sequential exposure to antigenically related influenza strains. So on the left, you see this person getting infected with influenza and its virus subtype A. Then subsequently, they get subtype A prime, and then subtype A double prime. And what happens in the body is the initial response to subtype A is recognized, you produce antibodies, and then when you get boosted or exposed to A prime, your primary response is directed against the first virus you saw, not the current one. And then similarly for A double prime. And this has been associated with negative outcomes because of the connotation with sin. I won't go there, but this is an important consideration. I'll show you why on the next slide. So basically, we all know that both antibody and cell-mediated immunity are important for viral control. If you look at the COVID antibody responses in the literature, they are very atypical. You simultaneously are developing IgM, IgG, IgA. About 75% of people make neutralizing antibodies. Sometimes those neutralizing antibodies don't last. And also, some of the antibodies decay very quickly. For example, with the licensure of the Abbott assay, which targets the nucleocapsid, at day 30, it has a sensitivity of about 95%. By the time 100 days passes, its sensitivity drops to about 71%. What is causing this? So there's some very interesting papers in Cell, Science, and Nature. And they're looking, in this case, at CMI and the T-cell responses being focused not only to the spike, which is that immunodominant part of the virus that's important for receptor binding, but also to other open reading frames. But they also find T-cell responses detected in non-exposed individuals. Next slide. So, as you probably know, there are 4 circulating human coronaviruses. There's 229E, NL63, no one's going to remember the numbers, but 2 of them, OC43 and HKU1, are beta coronaviruses. They are in the same family as SARS-CoV, and they have about a 70% genomic homology. And when your body is exposed to SARS-CoV, then the question is part of your immune response being driven by your previous exposure to these human coronaviruses, which have infected most of the population of the world? And so the question we have is, does SARS-CoV exposure drive an original antigenic sin kind of phenomena where the humoral and cell-mediated immune response is primarily directed at the past human coronavirus antigens and proteins versus COVID, which is what is hitting our population? And we believe that it is critical to understand SARS-CoV humoral and CMI to understand the pathogenesis and to help guide rational vaccine design and safety. Next slide. So this is a report from FUNC, from Frontiers in Pharmacology, and I'm not going to go into the vaccines that are out there. There are many, many, many different vaccines out there, as you know. If you want to know the latest, there's the London School of Hygiene and Tropical Medicine that as of September 4th, there were 241 vaccine candidates and 41 in clinical trials at the present time. Next slide. What you know is that in the US, you're going to have a vaccine before the election. And what would have taken 10 years from preclinical testing, clinical testing, phase 4 assessments is going to be compressed. And so the challenge for us is really understanding what is driving the immune response to this virus and whether or not prior exposure to other non-COVID coronaviruses changes that. Does it make the symptoms worse? Does it make the symptoms less? Does it provide additional herd immunity? Next slide. So in conclusion, infection leads to some protective immunity. We don't know the duration, and we're going to need antibody and CMI tools to measure it. If herd immunity is required, and 40 to 60% need to be infected to get that herd immunity, even in highly affected areas, there are many more people to infect, and you can see that with our rising cases in Canada and in other parts of the world, such as Spain, etc. The unintended consequences of our response are very severe, and we need to have civil societal discussions to come up with an acceptable approach, because shutting down society is not good for anybody. Finally, vaccines need to be safe, and typically they don't work well in the elderly, and everything is being compressed. So the better we understand the immunology, the better off we'll be. And finally, immune system, it works on a use it or lose it principle. There are going to be consequences to us living in bubbles and avoiding infections. So we need to come to grips with this in our context of our response to COVID. And I think I probably made it on time.
Thank you very much. That was really interesting, and I have a number of questions that I want to ask you. But before we get to that, I'm going to introduce our next speaker. For everybody listening, please go ahead and add questions to the chat, because I will be monitoring the chat as the next speaker is speaking, and I'll incorporate them into our Q&A. Our next speaker is Dr. Lewis Kraft. He's a research scientist at AbCellera within the Antibody Assessment Group. His research at AbCellera is focused on high-throughput antibody production, biophysical characterization, binding kinetics, and epitope binding. Luke has completed his PhD at the Sussex Drug Discovery Centre in the UK, and during his time there, he worked on a variety of early-stage drug discovery programs with a focus on neurodegenerative diseases. Anyway, thank you for joining us, and Dr. Kraft, the floor is yours.
Thank you. I'll be sharing my screen. Okay, I'll stop yours. There we go. Okay, can everyone see my screen? Or if just...
Yep.
Perfect. Okay. Well, thank you for the lovely introduction. So as you may be aware of, the drug and vaccine development nowadays takes a long time and a number of years, as also Mel outlined briefly in one of his slides. So that's why I'm very excited to be here today to talk to you about our efforts in combating COVID-19 and how we at AbCellera were able to identify a potential drug candidate ready for human testing in just 90 days. Now, before going into our discovery efforts, let's start with some background and also AbCellera's expertise. This will be a bit of a review for some people. So SARS-CoV-2 is the virus that's currently circulating in the population and that causes the disease COVID-19, with, as Mel outlined, also cases rising here in Vancouver. It's an RNA virus that primarily infects mammals, including us humans, and to enter a host cell in humans, for example, in the respiratory tract, SARS-CoV-2 uses spike protein to engage with the ACE2 receptor to enter the host cell to replicate and further propagate. A promising therapeutic approach, therefore, is to identify neutralizing antibodies that would block the ACE2 spike interaction and therefore stop viral fusion with the host cell and stop it from propagating. Such neutralizing antibody would be of immense value during this pandemic because it can be given to patients who are already ill and help them in their recovery. And it may also potentially be given as a prophylaxis in the most vulnerable population, so immunocompromised patients, elderly, etc. The question is, how do we find such neutralizing antibodies? And there are many ways to go about this. Now, we at AbCellera believe that nature makes the best antibodies, in particular antibodies that can be turned into therapies, for example, a therapy against COVID-19. In each human in their lifetime, there is the potential for billions of unique antibodies that the human body produces that are made by millions of different immune cells. So also in the case after SARS-CoV-2 infection, there's the potential for thousands of unique antibodies in a patient. The challenge really here is to see and screen through this immense diversity that nature has created. And this diversity is contained at the single cell level. So if someone really wants to find a therapeutic antibody with the ideal properties, it's about finding the cell that makes it. And we at AbCellera are world-class at doing that. We have developed in-house a very high throughput single-cell nanoscale screening technology that enables us to interrogate and screen millions of individual immune cells to look for those rare therapeutic antibodies, such as in the case of COVID-19. Now, beyond our technology, we also have expertise. So we have very strong expertise on ultra-hard, ultra-rare targets, such as GPCRs and ion channels. But we've been working on infectious disease for a number of years now. And part of our work has been funded by DARPA, that is the Defense Advanced Research Projects Agency, that is part of the US Department of Defense. And DARPA, prior to the current outbreak, initiated the Pandemic Prevention Platform, or brief, the P3 program. And goal of the P3 program is to stop a pandemic or slow down a pandemic in 60 days. So in detail, the goals of the P3 program in a pandemic scenario are to rapidly isolate a pathogen, for example, a virus, identify antibodies from a patient that recovered from the disease, identify those neutralizing antibodies, vectorize them into nucleic acid for manufacturing and delivery. And again, this whole process in a pandemic within 60 days to stop or slow down a pandemic. We at AbCellera are part of the P3 program, and we have developed capabilities towards that goal. So we have developed capabilities that could help to tackle pandemic threats like COVID-19. As part of the program, we also had to go through pandemic scenarios or stress tests to really test our capabilities that we developed. One of the first pandemic scenarios was against influenza, and the second was against the related coronavirus, MERS-CoV. So when COVID happened, we were ready to go. So how did our COVID discovery look like? We received a sample from one of the first US patients that recovered from COVID-19, and we were able to screen for neutralizing antibodies very fast from discovery into the clinic in just 90 days. So our screening or our discovery process started by receiving the sample, and it only took a few days to prepare and screen over 5.8 million individual immune cells against SARS-CoV-2 spike protein using our nanoscale assays. From that screening campaign, we discovered over 2,000 COVID-specific cells, so antibody-secreting cells that specifically secreted antibodies against SARS-CoV-2 spike protein. And we sequenced all these cells, and after bioinformatic processing, so that's day 9 of our pandemic response, we identified 440 unique human antibodies. Out of these 440, we prioritized a subset for rapid cloning, expression, and characterization. And we characterized these antibodies very extensively. So we characterized them in terms of their binding affinity, but also biophysical properties such as their stability, epitopic diversity. And with our long-term partners at the Vaccine Research Center, we also assessed these antibodies for their potency in neutralizing the virus. At this point, we also teamed up with world-class pharma Eli Lilly, who brought in a lot of expertise, in particular manufacturing and clinical expertise. We then collected and combined all this data into our in-house developed visualization software, Celium, and we used Celium to down-select our 24 antibody frontrunners. And those 24 antibody lead candidates were then passed on to the VRC, again for further potency testing, and to Eli Lilly for early manufacturing and clinical trials testing. So first human dose testing happened on day 94 of our lead candidate LY-CoV555 that already moved on to phase 2, phase 3 clinical trials. If you're interested, here are the IDs of the clinical trials. You can look up their current status on the trials database. And of course, with Eli Lilly, we are hopeful that the clinical trials will be successful. But taking a step back and looking at this discovery process, we were able to shrink a discovery process from what typically takes years to just a few months. And of course, we at AbCellera are hopeful that our expertise and technology that we developed over the last few years can be applied to other targets beyond SARS-CoV-2. And with that, I'm already at the end of my talk. This has been, of course, a huge collaborative effort that couldn't have happened with our terrific partners at the Vaccine Research Center, Eli Lilly and DARPA, in particular Amy Jenkins and her team at DARPA for their support and guidance in building the platform, and of course, everyone for listening, and yeah, also a big thank you to the organizers. And I'm sure Mel and I are happy to take questions now. I'll be stopped sharing the screen.
Thank you. Thank you very much. Very interesting. It's always I always enjoy hearing the AbCellera story. We're so proud to have you in BC and be a BC-based company. So I have there's a few questions on the chat which I'll turn to, but one of the things I want to start off with is you've both referenced speed, and COVID-19 certainly set the groundwork for speed. And I think what's interesting, Dr. Mel and I have been on a few panels together up till now, is I've often said we've been chasing science while we're also still discovering science and figuring out solutions. So I'm interested in what has been the biggest challenges to date in trying to maintain the balance between scientific discovery while you're still trying to chase it. Dr. Mel, could I start with you?
I think the hardest thing for all of us is the fact that everything changes every 5 minutes. Mm-hmm. So what you're seeing, and there's also the overlays of a population that is getting information from all different sides, and that information in BC may say X, and that information in Ontario or the US says Y. And so you have consumers who are confused. You have scientists who are publishing things before they're rock solid anyways, because anything can get published. And the complexity of what we're up against is enormous, because you essentially have a new virus coming into a population in a world where everything can be on Facebook or it can be on a Twitter account in seconds. And so this is truly a difficult challenge. I think also dissecting out what is true and what is noise, it has been very, very difficult. And so I just find, I think it's also relentless. And I think everyone is beginning to feel that in the public health sphere, because what does make sense? And at the same time, we know we need to get kids back to school. And you have a lot of parents who are nervous about their kids going back to school. So I don't know if I've answered your question, but this is a tough time for everybody.
Mm-hmm. Mm-hmm. Lucas, did you want to add to that?
Yeah, I don't have really much to add to that, but for sure, I think maybe out of my personal perspective, of course, we are in this pandemic all together. We are all, learning on the go. So many new things about this virus. It is, like Mel said, it can be really hard keeping up with the literature and what is, truth sometimes and what is noise. I think that is really a challenge currently in this pandemic. But I mean, unfortunately, it's also very exciting, all the things that we are currently learning about this virus and, the immune response, etc., and how we can actually tackle this pandemic. Yeah.
And I mean, I think it goes without saying that this is super exciting from a science perspective and discovery perspective. And if time was on our hands, it would probably be, a career like exciting opportunity. But there's also, as you're both saying, this pressure of timing and this pressure of maintaining the degree of scientific discovery and integrity in that discovery that we all have come to enjoy, especially in Canada, where we really keep a high bar on the quality of products that end up coming into our healthcare system. And so I wonder, and Dr. Mel, you just referenced it, there is so much information out there, and it's coming from different jurisdictions, both globally and within our own countries. How do we maintain the public confidence in bringing these discoveries into the marketplace, whether it be a vaccine or a therapeutic? Because all eyes are watching on these first therapeutics and vaccines that end up being utilized broadly, and there are risks associated with that. So how do we try and make sure we find that balance between the timeliness that we're all looking for at the same time as maintaining the quality and integrity of the solutions?
So one thing I will say is that it's hard to imagine the technology we have now versus the technology we had 10 years ago. You would never have been able to have a vaccine out within a year or two 10 years ago. And the technology that Lucas presented is just outstanding. And I think the importance is to try to use as much scientific rigor that you can, and also to be transparent with what you know and what you don't know. And I think it's critical that we do that as scientists, but it's also critical that we say it to the public. I think that if we try to protect the public by saying we know this and we don't know that, they want a straight story, and I recognize that that straight story is not simple.
Mm-hmm. Lucas?
Yeah, I think what Mel just mentioned is technology has changed over the years. I mean, by all means, my scientific career is probably not comparable to Mel's or other speakers here. I'm still a young researcher, but yeah, I think it has changed. I think we are in a position to really change the drug discovery process, to really shrink something that typically takes so long to a shorter time frame, given our technology that we have. And yeah, I think we can learn so much from a pandemic like this, and apply that maybe onto future projects as well and other drug discovery efforts.
Mm-hmm. So I'm going to turn to the chat because we have a couple of questions. I assume you're going to wave at me when our time is up, Kara. Okay, so Dr. Mel, this is for you. Is it known how many viral particles are being required to actually detect the virus in the diagnostic assays, and how many virus particles are required to become infectious? And can this be monitored?
Yeah, that's a good question. So one of the things is important to know that when you're measuring RNA from a patient sample, that much of the RNA represents defective products and not a fully replication competent product. What we do know, and we measure what's called the cycling threshold, how many cycles you require to detect it. And there's literature that suggests that samples which have low viral quantities, typically in the range of cycling thresholds of 24 and above, tend not to grow in cell lines. And when you look at that compared to an antigen test, here you're looking at proteins, and typically protein detection is about 10,000-fold less sensitive than PCR, but the detection of antigens probably correlates better with actual replicating competent virus. The challenge for us is really being able to recognize what you can do in vitro, because the cell lines don't necessarily replicate what's in your nose or in your lungs. So it's this challenge, which is, you're absolutely correct, but somewhere along the line we can make some guesstimates, but we can't be very precise about what that number is.
Right. And I want to ask you, Lucas, so you guys have, I mean, it's extraordinary what AbCellera has been able to accomplish and how far along you are, and as you referenced, it's, you know, rapid action and it's quite a shorter time frame. So based off of the clinical trials where you are today, what is the expectation of, you know, if they're going well, that a therapeutic may be available?
Expectation, sorry, do you mind clarifying?
Of timing of when they may be available.
Oh, that is a very good question. I mean, again, like everyone here, we are all sitting in this pandemic. We are all looking for, you know, for a therapy, you know, as soon as possible. Eli Lilly is currently conducting the clinical trials. I myself don't have visibility on their clinical trials and timelines. I can only say, you know, follow, you know, Eli Lilly's website or our website for like the latest details. Yeah,
and it would be a hard question to answer anyway. And while Eli Lilly is doing those trials, are you continuing to look for other potential solutions to those trials not occur?
Yeah, we are very focused on our COVID efforts still, and we are prepared to look for additional therapies, yes, moving forward.
And with the DARPA partnership that originated, is there a group that are continually connected through that program in the discovery process?
We are, yes, we receive a lot of support from DARPA, and as I mentioned before, in particular, Amy Jenkins and her team who really, you know, gave us support and guidance in building the platforms and capabilities.
Okay, great, thank you. Are there any other questions from the floor? Otherwise, I think it's time to move to the next panel.
I'm just seeing actually a question in the chat by Joseph, I think, about, if that's okay if I can take that, or should we move on? No. Okay, I'm just seeing spike is heavily glycosylated, and which may pose a problem in antibody discovery. That is true. I mean, spike protein, I think, is a super fascinating protein, and I've been looking at that in very detail. I have a structural biology background. What I can say is there are publications that actually also show, although spike is highly glycosylated, in certain conformations, in particular the up conformation of the receptor binding domain, it's actually not very glycosylated, which enables also excess of antibodies. I am not sure if that answered his question, but yeah.
Well, thank you for picking up on that.
Thank you.
Okay, there was one other one that we can maybe address through the chat. Okay, over to you, Kara.
think I'm supposed to pick up here, is that right? So it's Deborah Money here, and I'm more than happy to moderate the second half of this very interesting afternoon. And we have two excellent speakers coming up. First off, I'd like to introduce Dr. Daryl Knight, who is currently the Vice President of Research and Academic Affairs at Providence, and also a professor in anesthesia, pharmacology, and therapeutics at UBC, and Associate Dean Research for the Faculty of Medicine. His background is he obtained a PhD from the University of Western Australia as a cell biologist with an international reputation in the area of lung and airway modeling. And he's going to talk to us about COVID-19 from his perspective. Daryl, thanks very much.
Thanks. Thanks, Deborah. I'm just going to share my screen if I can find it. There we are. Can everybody? I've just got to get onto a the slideshow. Everybody see that?
We saw it briefly, Daryl, but now we don't see your slides. Do you want to try again?
Oh, okay. Can you see, Dave? I got the thumbs up from you, so.
I can see it too, Wendy.
Oh, okay, I'll try again.
This meeting is being recorded.
Can we see that?
Not at the moment, no.
Okay.
You were up briefly.
I could see it the first time.
Now it's gone again. Interesting. Hang on a second. I will close that.
Kara, does anyone have his slides at your end? Backup option is they could show it and you could just say, direct them to move slides if you want, Daryl.
That might be the easiest thing because it was there and now it's gone.
Kara, anybody can put his slides up?
Jared, do you have the, did you send your back? Oh,
Looks good, there we go.
Great. So I don't have control of it, so I'll just have to ask you to click, Kara, thank you. So just go back one slide, if you may. Thanks. So Deborah, thanks again for my introduction, and thanks to David, Amanda, Vivian, and Kara for setting up this great series of workshops. And I'm delighted to be given the opportunity to present at the inaugural one and share this virtual stage with the eminent thought leaders that have spoken before and after me. So thanks very much. Sorry, go back one. My brief was to talk about COVID, collaborative research post-COVID with a focus on biobanks. I've sort of expanded that brief a little bit just to include a few other things. I want to highlight that biobanks, to me, are the key and core component of what I'm about to talk about. So I'm not glossing over them. I just think they're a key part of a bigger ecosystem. So next slide. So what I would like to talk about is moving our thinking from silos to ecosystems. And I think that as hopefully as this talk goes on, we'll get an understanding of how siloed research has only taken us so far. And in the new normal, perhaps we need to be thinking in a larger and more collaborative fashion. Next slide, please. So I don't think there's any doubt from the talks we've heard or your own personal experience that COVID's had a dramatic and disruptive effect on our personal lives and our work lives. But I think also COVID's also been a phenomenal social experiment. And from a research perspective, I've been amazed at how quickly the research community in Vancouver has responded and pivoted to tackle this pandemic. And I've just highlighted a few of the initiatives that have come through in the last 3 to 4 months. We've seen significant increases in targeted funding for COVID-related research from a multitude of local, provincial, national and international funding bodies and enterprises, which has been absolutely rapid in its formation. From a VP's perspective, it's great to see reduced bureaucracy, and we've also seen heightened engagement and productivity from industry and from the government. Last year, if you said we were going to have a vaccine developed in under 2 years, not many people would have believed you, considering that it takes generally well over 10. And I think our universities and the provincial government, with the formation of their task forces, has been absolutely brilliant at enabling multi-site collaboration at scale. And what that has meant, though, is that we have already generated substantial amounts of innovation. We've seen improvements in clinical testing, we've seen fluorogenic testing, less invasive testing, more rapid turnarounds in getting positive or negative answers, which has been an amazing opportunity for the patients. We are developing, I should say, the BC COVID Biobank Network, which was going to fit hand in glove with the Interdisciplinary Clinical Care Network, which in itself is an innovation because it's a new way of treating patients who are presenting with comorbid diseases. So I think there's a number of initiatives that we can really focus on and take advantage of as we move forward into the new normal. Next slide, please. So I think we can all agree that we're past the old normal. We're not going back there. The bigger question is, what do we do when we get to the new normal? And what does the new normal look like? And then next slide, please. And even after that, what's beyond the new normal? And how can we use what we've learned through COVID to better support discovery and commercialization in Vancouver and in British Columbia? We all know, those who have been exposed to it know that innovation and as a consequence commercialization faces a multitude of hurdles, particularly when universities are involved and their relationships with industry and government. And one of those hurdles, I believe, is linear transfer of information, which is wholly inadequate for what we're talking about now. I believe that in an ecosystem approach, a network of networks, if you will, which are mutually reinforcing, will support and enable more rapid commercialization of those innovations that we've been pursuing. Next slide, please. Moving in this direction, however, is going to take a change in mindset for quite a large proportion of the population. But I do want to stress that this is not about fixing systems that were never meant to work together, nor is it about trying to break down every barrier and fence that we can find. Rather, what I think the opportunity that COVID is providing us is a way to develop new, more productive, collaborative relationships and the solutions that will follow from them. Those relationships, however, must be predicated on inclusiveness, of open-mindedness, of knowledge sharing and respect, or else it's not going to work. And I want to keep referring back to that because for an ecosystem approach to work, we need to be able to trust and respect the work that each of us is doing. Next slide, please. This is not a new initiative by any stretch of the imagination. We've been talking about this in my previous visits in Vancouver. And the federal government, as early as 2015, was saying, if we can do it within a province, we've got to get better at integrating where we're going to spend money and where we're going to see the benefits. The provincial government, every opportunity they get to talk to myself and others, are telling us, work together. We need to work together if we're going to attract ministry funding and ministry attention and ministry support. So I think we've got the mandate. We've been given the impetus through COVID. We're at that time now, what we're going to do with those two things force feeding us into a direction. Next slide, please. So what's a model of a BC biomedical innovation ecosystem look like? Well, I think it's going to be different for different people, but hopefully we can coalesce around a fairly defined approach. For me, it's about patients and the communities and the patient support networks. Then you mix in the government, research, spin-offs and startups, hospitals, biotech and pharma and research universities. And you coalesce around the people in the middle to answer questions that are important to them and are priorities for the health system. That's what I mean by a biomedical innovation system. How do we get there is the bigger question, and I just want to provide you a snapshot of what I think our first step is. Next slide, please. And for me, it's building a shared integrated platform for supporting the innovation and research that we are currently pursuing. And Dave Granville mentioned this at the start about an asset register. This is something I think that is key to the innovation ecosystem in Vancouver. If we have an idea of what some of the technology and infrastructure exists within Vancouver, how we can best share that equitably, and how we can best promote the research that's going on within the network, then I think everybody is going to win and research will be advanced at an accelerated pace. Two examples that I think we can learn from. One of them is the SciLife Labs. This is in Stockholm, just adjacent to the Karolinska Institute. And it's 5 floors of essentially core facilities that's supported by the philanthropic family, I think the Wallander family, if I remember correctly. This is the home of the Human Protein Atlas, amongst other things. And those core facilities are available to any researcher in Sweden, and I think throughout Scandinavia, at cost. And there's technical expertise, there's variety amounts of research input that's based on your level of comfort of using the equipment. Next slide, please. If we take it one step further, no, just go back one. Thank you. One, there we go. The European Union has a network of core facilities. This is called Core for Life. And each of these core facilities, they all have similar technology which is available to all researchers, but they also have specific individual areas of strength, which if you want to use a specific type of equipment that's not available, you are able to send your samples there, and it will be analysed, investigated to your expectations. So there's 2 different extremes. We've got a building, we've got a network of buildings. What can we do in Vancouver? Next slide, please. Thanks. Well, hopefully you can see that is our little network of research universities and hospitals. You can see that they've all got dashed lines running between them in a bit of a spider web. That green dot you can see is where the new St. Paul's development is going to be in 5 years' time. But we have an opportunity to do a bit of a hybrid. We can share the infrastructure that we've got in our various institutions, but make it available, open. More importantly, moving forward, share the intelligence of what we're going to buy so that there's not duplication where we don't need to. Most of us in the research world have spent enormous amounts of money on a piece of equipment that was used for 6 months to a year, then gathers dust in the back of the lab. That's something we can't afford to do moving forward. And I think the best way to go about to stop that happening is to develop this shared network. Next slide, please. So what are some of the other systems? Well, obviously I've talked about research networks within networks. Data is probably one of the strengths of our province. And the federal government health and bioscience strategy table identified data as a key barrier to innovation. Now, I've got a quote here, I can't remember it, so I'm just going to read it out. They say that a disconnected digital health system for the collection of data to inform decision-making and advanced scientific innovation is a key barrier to innovation. And they concluded that a recommendation for Canada to develop fully operable digital health platforms across jurisdictions. So again, we've got a mandate. For me, data is important, but it's half the story. We need linked biobanks linked to the patient data to generate the full picture of understanding disease pathogenesis. We're starting to get closer engagement with the provincial and federal government We need more engagement. We need more funding. We also need them to enact different legislation, have a say on regulations and policy that are going to support innovation and not stifle it. And as I said, above all, we need trust, motivation, leadership, and support. Next slide, please. So what have we learned today? You can just click, I think, 3. One more. Thank you. So COVID-19's been a brilliant galvanizing force. It's highlighting the benefits of a cohesive link between research that's guided by and informs clinical activity. It highlights the significance of a multidimensional approach, including biobanks, including clinical trials, and including well-curated data. And it also highlights that we need this open intersection between government, industry, healthcare, and research. And final slide. So I know we're all fatigued by COVID and we all wish it was over. Unfortunately, it's probably not going to be for a little while. But we are approaching that point in time where we have the opportunity to explode further and develop really, really strong ecosystems, or we don't. And so my question to everybody is, are we going to maintain the rage? My hope is that the answer is yes, and I'll stop there and take questions when it's appropriate.
Daryl, thank you so much for that excellent talk. I think the format is the same as before, that we're going to do the 2 talks and then do questions after, but I'd encourage anyone with questions for Daryl to pop them into the chat, and we will get to them after the next speaker. So it's my pleasure to introduce Dr. Josef Penninger, who I'm sure is well known to many. He's the Canada 150 Chair in Functional Genetics. He's the Director of the Life Sciences Institute at UBC. His background is that he studied medicine at the University of Innsbruck in Austria, and one of his accomplishments, of which there are many, was that he was the scientific and founding director of the Institute for Molecular Biotechnology at the Austrian Academy of Sciences. He's had significant success in his research career, particularly notable in this context, demonstrated a critical role for the ACE2 as a cellular receptor for SARS coronaviruses in the initial SARS outbreak, and has linked ACE2 to link that to lung failure in such infections. He's extensively published and internationally renowned. So, Josef, thank you so much. Please go ahead.
So, yes, thanks so much for having me and to present my little story today. And also thank you so much to the other speakers. It's spectacular research, very important research, and also Daryl, you talk right out of my heart because I actually believe only with common infrastructures we can be really successful, and actually also believe there's such waste going on if we don't do this. And actually the secret of success of my place in Vienna was to do exactly this. So I would really wish to move forward, and of course together with everybody. So I want to tell you about the story and also the data which are coming out today in Lancet on the first clinical use of our medicine. So the story started, we were interested in fly heart development and realized that flies had actually 2 copies of ACE. This was in 1989. We thought the flies have 2 copies of ACE. Of course, ACE is a regulator of the renin-angiotensin system, which had been known. Then mammalian should have 2 copies. So Mike Krakauer, a kid from Toronto in my lab, cloned ACE2 angiotensin converting enzyme number 2, like ACE, a transmembrane enzyme which cleaves small peptides. I should say we were not the first group which published the sequence because I was always in knockout mice and in vivo function. So we published the first knockout mouse in 2002 in this paper by Mike. And basically the first phenotype we found was impaired heart function, which of course it becomes really interesting in terms of COVID-19. Over the years, my group and of course many other groups, and Mike's paper was really the defining paper to show that ACE2, the critical in vivo function of ACE2, is actually to reduce angiotensin 2. ACE makes ANG2, ACE2 gets rid of Ang2, and Ang2 via two G protein coupled receptors regulates blood pressure, heart function, so kidney disease, fibrosis, blood vessels, thromboembolism. This even drives inflammation, drives IL-6 production, for instance. Many aspects we can now, of course, associate and have to associate with COVID-19. But I also want to mention that ACE2 is not specific. It can cleave other little peptide systems like bradykinin. And as you know, there's now this new ideas coming out that bradykinin might be involved in leaking blood vessels, contributing to the severity of COVID. There are other peptides like apelin and neurotensin. So ACE2 is the critical negative regulator of the renin-angiotensin system and also affects other peptide systems. So we started very early on, before there was actually SARS on the planet, to test the role of ACE2 in acute lung injury. A postdoc of mine, Yumiko Imai, with Slutsky in Toronto, had developed over many years a model for intensive care in mice. Probably only 3-4 groups in the planet working on this. It's very difficult to have these models have very reproducible systems. And it turned out when we mutate ACE2, lung failure became much, much worse. So Yumiko basically established a new paradigm of ACE2 function in the renin-angiotensin system in protecting the lung from acute lung injury. And then SARS came along, and Mike Fasan at this time in Boston had this paper in Nature saying that the spike protein of the SARS coronavirus, this was in 2003, would actually use ACE2 as an entry point for viral fusion and to get into the cells. But very early on, there were many other candidate receptors, like lectins, and this was also where my question came from of glycosylation of SARS-2 spike, because heavily glycosylated, so it binds to lectins. And so the question was really, is ACE2 the essential receptor for the SARS coronavirus? Since we had the only knockout mouse in the world at this time, we sent it to China. The Chinese infected it with mouse-adapted SARS coronavirus. And as you can see, wild-type mice, we can recover infectious virus, and there's some minor lung disease. And if we do this in ACE2 knockout mouse, there was no virus. So ACE2 is the critical in vivo receptor for the SARS coronavirus. And we also could explain mechanistically in lots of experiments, which we published in 2005, why this particular virus might have become a killer virus. It actually spikes, binds to ACE2, and this leads to downregulation of ACE2. So ACE2 is lost from the surface of the cells. However, unspecific lung injuries like virus injuries, pneumonias, sepsis, acid aspiration, turns on the renin-angiotensin system, turns on Ang2, which drives severe lung injury, and we map this all genetically. And of course, if ACE2 is down, then Ang2 becomes dominant, and the injury becomes worse. So the idea was, if this is real, then basically lung injury in part, is because of reduced ACE2 expression, and then we just supplement the system with a soluble recombinant form of human ACE2. This was our first experiment. This is lung injury. In our mouse model, this is acid injury, changed lung functions here in acid control, and when we give you recombinant human ACE2, we can significantly alleviate acute lung injury. So based on these studies, a biotech I founded to declare my conflict in interest in Europe, developed human recombinant ACE2 with the name APN01 for therapeutic use in ARDS, acute respiratory distress syndrome. And there are many reasons which actually leads at the end of the disease to this severe lung failure phenotype from flu, Spanish flu, to bird flu, to anthrax, bioterrorism, and of course the SARS coronavirus. So we labored away. We actually worked together for some years with Glaxo. We tested this drug already in phase 1 and phase 2 clinical studies. In total, 89 humans healthy volunteers, and in phase 2, people with lung injury and then pulmonary hypertension actually got the soluble version of phase 2 with a reasonable safety profile. So it's an IV infusable soluble form of phase 2 with a half-life of around 12 hours. So this was nice and was really nice work on mechanistic understanding why the SARS virus became a killer. But of course, then the SARS virus disappeared and the relevance was more or less gone, besides of course developing the drug for acute lung injury. And then the new SARS virus, SARS-CoV-2 emerged. And then the sequence was published, as already pointed out by the speakers before me. It was very clear because of the spike. Here's some sequence data, so some structural data, and in green is ACE2. That ACE2 must be the receptor, the critical receptor, so not excluding there might be minor receptors around, but ACE2 must be the critical receptor. This is our own modeling, which is submitted with in red here, all the sugars. So actually there are lots of sugars in the interface. There's actually 40% of the RBD is covered by sugars and 60% of the interface of ACE2 is covered by sugars. So this will be very important to design rational drugs, but this is another story. So it was clear ACE2 is a critical SARS-CoV-2 receptor. So then the question was, can our soluble form of ACE2, which we had been developing for more than 10 years in the preclinic for safety testing, in human volunteers and in phase 2 trials, could this actually be used to stop the SARS coronavirus infection? And of course the idea is this: if this virus binds to ACE2 and uses ACE2 to get entry into the cells, this leads to internalization of ACE2 together with the coronavirus 1 and of course the coronavirus CoV-2 causing COVID-19. And inside the cells, of course, then the virus does what it's doing, spills out, replicates its RNA, makes its protein, interferes with the immune system of the cell, at the end causing diseases we call SARS and COVID-19. And this was the idea of the soluble version of ACE2 as a decoy, functioning basically what you already heard from Abcelera as a soluble neutralizing molecule because it of course had super high affinity, 1.2 nanomolars affinity for the spike protein, and of course if you get soluble version it would block the virus finding its membrane-bound receptor, there should be less infection and of course improve disease. So this is probably the principle of 50% of drug developments going on at the moment for COVID-19. From vaccines, from antibodies being developed, from small molecules being developed, interfering exactly with this interface between spike and ACE2 to block this binding of the virus to ACE2, so to block entry. So to do this, we actually hooked up with the Karolinska Institute in Sweden, the high security lab. So that's the virus we used. It was the virus isolated from the first patient in Sweden. And we infected the ROE6 cells with the virus here in the black bars, and then we gave APN01, so the soluble form of ACE2. And as you can see, we can reduce the virus load in the cells in a dose-dependent fashion by a factor of 1,000 to 5,000 times. If we use mouse ACE2 on the bottom here, it doesn't do anything. It was a great control because mouse ACE2 cannot bind or to minor binding to the spike and the SARS coronavirus 2. The second issue we wanted to address is this. So how does one explain actually COVID-19? It starts in the nose, gets into the lung, but then at the end it's a multi-organ disease. And ACE2 at least can explain the tissue tropism of the infection because ACE2 is heavily expressed in the heart. I showed you our first phenotype. It's expressed in blood vessels, in the nose epithelium, in the central nervous system. It's on the X chromosome, so ACE2 expression might change with gender, cardiovascular disease, diabetes, smoking, change ACE2 expression, as you would expect in a system which is critical involved in health of the blood vessels, regulating blood pressure, which is, of course, very fine-tuned. We also published papers that ACE2 is in the intestine. COVID-19 patients, in many cases, have diarrhea because we can get access there. And it's also in the kidney here in green, a heavy ACE2 expression. So can ACE2 explain multi-organ COVID-19 involvement? And this is, initially it was really a lung disease, it's pneumonia, and then there's the cytokine storm happening and the overshooting immune system, and of course this would lead them to damage of other organs. So we wanted to show if the virus can directly infect these other tissues, knowing that ACE2 is expressed. So to do this, we hooked up with Nuria Montserrat in Barcelona. She engineered human kidneys from stem cells. So this is how they look. The green cells are actually the ones which have ACE2. That's the proximal tubules where ACE2 is expressed. And sure enough, if we infect this tissue organoids, and the whole field is now exploding, people using organoids in many variations to study COVID-19. So this was really the first study and the publication doing this with Nuria. So when infect this kidney organoids with the virus, then we can recover viral RNA, we can recover viral progeny, so they actually make new virus which can infect other cells. And again, it can be blocked with the human recombinant soluble ACE2, as you would expect if ACE2 is important for entry. The other thing we did was we did blood vessel engineering, which we also do actually with stem cell technologies here in Vancouver, which is really great to work with them. And we developed last year actually blood vessel capillary organoids from stem cells. So this is how they look. We infected them again. And as you can see, we can recover viral RNA, viral progeny, infectious virus, and again, it can be blocked with So this all works, and clearly the virus can get entry to these tissues, so it's a direct demonstration, and of course the pathologists have shown and can find virus in kidney, in the heart, in the intestine, in the blood vessels. And so the disease starts in the nose because there are lots of ACE2. In the throat, there's lots of ACE2, so the droplets or aerosols can get access. So for most people, probably it ends there, or they might not even get the disease. If it gets deep down into the lung, ACE2 is in particular alveolar epithelial cells. That's why the patients get the very typical deep and very severe pneumonia, and of course pneumonia leads to damage, tissue damage. The virus can spill out, gets access to the blood stream, can infect endothelial cells directly, can spill into tissues and can pass on. So this clearly the virus can do this. We believe it's an important part of the pathology. Of course, there's cytokine storms, there's direct viral damage of the happening, and of course many other aspects we have to consider. So this is basically what I said, this multi-organ involvement, so COVID-19 is initial, in most cases, lung disease. Of course, you'll always have this case of strokes or thromboembolism without having much lung involvement. But in most cases, it's a severe lung disease in the severe stages of the disease, which is then spilling out in other tissues. So late stages of multi-organ failure and multi-organ disease. That's probably why it's so difficult to treat.
So Joseph, this has been absolutely fantastic. We're just a little tight for time for questions. Are you just?
1 minute,
can you wrap up? Thanks so much.
So this was the idea that we're doing a placebo double-blinded phase 2 trial for severe COVID-19 patients with these molecules. The idea being it reduces infections at the same time because of the function of the renin-angiotensin system protects multiple organs from damage. So today, actually, we published the first clinical use of the molecule in a patient. And this was very important. So our clinical trial is still ongoing, but we needed to know what actually happens in a real COVID-19 patient, because there could have been some issues. So is the enzyme function still operational? So this is basically exactly what we show here. So this is angiotensin 2, so here in red, and if we give ACE2, so this is a patient, a woman who had severe COVID-19, who is now healthy at home again. so the high angiotensin II levels. If we give ACE2 within minutes, angiotensin II goes down as one would expect from an enzyme functions, and angiotensin 1 to 7 and Ang 1 to 5 go up as one would expect from the enzyme function. This is day 5 of treatment, so day 0 is before treatment, in 3 to 7 days. And if we stop on day 7, you see Ang2 is still working, and day 11, actually it comes back to the baseline. So the enzyme function works in a real patient. Secondly, the cytokines, the inflammatory cytokine storm goes right down. So in red, we start treatment twice a day for 7 days. In green, we stop it. You see IL-6 is shooting down, IL-8 is shooting down, RAGE is shooting down, ferritin is shooting down. Some other aspects are not so clear, but they're still working on this. And the third was very critical, is what actually happens when you come with a molecule like ACE2, and also with antibodies we heard before, neutralize the virus. Does the immune system still start a proper immune response against the virus? Because it could be that the immune system cannot see the virus anymore. And this is what actually happens. So again, start of treatment is the viral load, as you can see in the blood, goes down rapidly under detection. All this is plasma, also in the trachea it goes down, and nasopharyngeal swabs it goes down, and the patient developed high titers of neutralizing IgM, IgG, and IgA antibodies. So as said, the paper is actually coming out today, being the first ever reported clinical use of ACE2 in COVID-19 patients. And wow. With this, thanks for listening, and many people who helped us. I want to mention Alexander Zufali as a clinician back in Vienna, who actually treated the patient for the compassionate use, and deserves really the credit for this first publication. I think everybody was waiting for, because the whole field was expecting what is ACE2 actually really doing, can this be really used? Now we know it can be really used, and of course the clinical trial will see in many patients if it's clinically efficacious. So thanks for listening.
So Josef, thank you so much for presenting this very exciting work, and we're all, of course, really interested to see the outcomes with this novel agent with larger clinical trials. Very exciting and promising. We have just a little bit of time left for questions. There's been a few in the chat, that probably more than we can get to, but maybe I'll just flip back to questions for Daryl. And there's a couple of questions really on a similar theme around this issue of you suggesting these integrated core platforms, these networks as opposed to silos. Essentially, do you have more sort of specific suggestions as to how we do it? There's one question about the fact that we've got genomics in Vancouver, proteomics in Victoria, metabolomics in Edmonton, for example. How do we realize this in Canada where they have been able to realize it in Europe?
Thanks, Devin. I think Wendy actually might be able to chime in on this too, but we had an earlier call. Life Sciences BC has done a similar type approach to what I'm about to suggest. The first step we need to do is develop an asset register to know who's got what and where it is. So a great example of this, I spoke to Colin Collins at the Prostate Centre a couple of months ago. They've just spent $2.5 million on genomics equipment, which is great because it means we don't have to. And they've said they've opened it up to everybody to use. It's just there on an equitable basis, not on a Sunday after midnight type thing. So, I think that's the first thing we need to do is need to know where things are and who's got what. The next step is to share intelligence around what's going to be best if we need a multi-million-dollar piece of equipment. What's going to be best for the group? So we don't want to stop people going out and getting pieces of equipment that are going to be useful for their own research, but we also need to come together around super pieces of equipment like the $600,000 to $700,000, $1 million pieces of equipment, and really go all in as a collective. So the first step is that asset map. The second step is to what Dorota was talking about. Link with Genome Canada, Genome BC, to see where we can share equipment again. And the whole point of this is not spending money when we don't need to, and making the shrinking pool that we're getting go further. And also getting the value add from working collectively and working together. The Life Sciences Institute would be a great house for, for imaging and for all sorts of things. And I know we've been discussing that, but I think we've got to start acting on it and really trying to understand what pieces of equipment are going to work really well where, knowing that it may not be in your institution, but you still have equitable access to it. So for me, that's the first thing. The other question about, support, the government needs to support it.
I was going to ask about that next, Daryl, not just about the linked core facilities, but, the legislation and so on that you referenced. It's something that I'm up against quite a lot right now in the work I do that across the country, where you come up against these barriers that are related to sort of essentially archaic rules.
Yeah, and I think we've got to use, this is why I keep saying, I know we're tired, but if we can't do it with COVID backing us up, we're never going to be able to. So, I already understand that we've been temporarily able to park data outside of Canada. So I mean, we can do it. It's just how do we keep it going? And so it becomes the government's new norm. The same with the universities. I think they've got to step in and bat for us. They've really got to come to the party, and Santorono, Dermot, Joy Johnson and Tanya Bugler need to come to the collective and put their weight behind what we're trying to do as well.
So thank you, Daryl. Thank you, Joseph. David, I'm just doing a time check. Do you want me to stop here, or is there do you want to go on with any other questions?
You're on mute, Dave.
I think at this time, I think it would be best to, I'm just going to share the final slide here. I think it would be best to wrap it up and thank all the speakers. Going through, this was a great session to finish off with. The talks were outstanding, and you think about the size of Vancouver and the scientific community and how we're batting above our weights. You look at we're a role model around the world when it comes to managing COVID. We heard today, not only thanks to Mel and Bonnie and the rest of the team at the CDC, you look at today what we heard. We got one of the top people in the world who actually discovered the receptor for the virus. We then saw a company which was able to take something from start to finish into phase 3 clinical trials, which is virtually unheard of. And this company's also from Vancouver, and I know they're going to be recruiting a lot of people in the future, so we hope they remain in Vancouver. And then lastly with Daryl is, of course, this has been happening, but there's so much more we can do with our technology once we all start putting our minds together. So I'd like to thank all the speakers, and then once again thank Kara, Vivian, and Amanda for the great job they did organizing this. And just to quickly go over the next date, to save the date, we have an October 29th meeting on digital innovations, November 26th on medical devices. We're looking for suggestions, so in the feedback forms, please give suggestions. And again, what we want is a clinical input, scientific or engineering or computational input, and then a couple companies that are addressing the issue, and we can come up with one of the core facilities that we'd like to advertise or promote in the session as well. So with that, I thank everyone, and have a great rest of your day. And thank the moderators as well, Deborah and Wendy. Thank you so much.
Thank you. Great session.
Thank you for putting such a wonderful afternoon together. Well done, everybody.
Yep.
Thank you, everybody.
The recording has stopped.
You are the only participant in the conference. Your call will be disconnected.